بسم الله الرحمن الرحيم. We always know that there is a variation of human features. For example, the features of an Egyptian are different from Africans, both are different from Europeans or Asians. Parents pass their features to their children, so the children look like their parents, and we can identify them if we look at them with their parents. And we say these features are due to inheritance of the characteristic features of the parents. Now, what is inheritance? Actually, the science that deals with the study of inheritance is called genetics. The scientific study of inherited human variation is called human genetics. In human genetics, we deal with the study of the mechanism and the rules of transmission of inherited traits through generations. What do we mean by inherited traits? These are the characteristics of a person which could be either features like eye color or tallness, or diseases and disorders such as Down syndrome and cleft lip and palate, or metabolic activities. For example, the inability to process galactose, a monosaccharide found in milk sugar lactose, is called galactosemia. This leads to brain damage, cataracts, jaundice, enlarged liver, kidney damage, and so on. Now, how are these traits are inherited, and what controls this inheritance? Actually, the human genome is the complete set of genetic instruction of these traits. It is found in the DNA of the nuclear genome. The DNA of the nuclear genome is found as a sequence of 4 nucleotides. We can say that the nucleotides are like any letters, the letters of any language, and the sequence of these letters gives certain words which have a meaning, and so the sequence of the nucleotides in DNA gives the information of genetically inherited traits. Nuclear genome includes coding genes, and these coding genes are the parts of DNA which controls the appearance of the trait, and non-coding genes, and these non-coding genes actually work together with coding genes in order to control the time, the amount, and the extent of the material which is formed by the coding genes. Another gene is found in the form of DNA in mitochondria. DNA of mitochondria represents the mitochondrial genome. And so, in the human cell, we have 2 types of genomes: the nuclear genome and the mitochondrial genome. Both of them actually control certain traits so that we can identify these traits and the relationship with nuclear and mitochondrial genomes, as we will see later. Now, how is the information in DNA inherited, and how does it controls the appearance of a trait? Inherited traits are performed and or directed by genetic information in a specific gene. Maybe one gene or several genes, but the gene has the information, as we said, as a sequence of nucleotides. And this gene is a part of the genome which has many other genes controlling other traits. Now, the information in the gene in the form of sequence of deoxyribonucleotides of DNA are transcribed into a sequence of nucleotides in RNA. And we will know that there is some chemical difference between the nucleotides of the DNA and RNA, but we have to know that the sequence of nucleotides in DNA is found in the genes and the genomes which are found in the nucleus. But when it is transcribed, it forms the RNA which can move from the nucleus, as we will see, to where it can be processed in the cytoplasm of the cell. The RNA leaves the nucleus, and in the cytoplasm of the cell, the sequence of nucleotides of the RNA are translated into a sequence of amino acids in the protein. The protein actually, as we will see, is the product, the gene product. Maybe the protein by itself, such as the unusual collagen or the collagen which is not formed properly, leads to the formation of stretchy skin in Ehlers-Danlos syndrome. Or the protein can be an enzyme or several enzymes which control the processes, certain processes, such as the formation of melanin, which is the black pigment, which when found in a large amount in the skin, the human is black colored, and if it is found in a small amount, the human is white. Or it can be in the form of hormones, such as the case of growth hormone deficiency, which leads to dwarfism. And so the direct product of the gene is a protein which can act on certain parts, as we'll see, in order to show the traits. Now, the human chromosomes, when nuclear genome is transmitted from parents to their children, or from a cell to a cell during division in order to give two identical cells, we'll find that it is not transmitted as DNA, but it is packed into chromosomes. The chromosomes in human, the nuclear genome is represented by 24 single chromosomes. These 24 single chromosomes are of two types. The first is 22 chromosomes, which are called autosomes, and one pair of chromosomes, which is X and Y chromosomes, sex chromosomes, which controls sex, as we will see later. Okay, so we have autosomes that carries the genetic information of all the traits of human being, and the X and Y chromosome, which together with carrying certain information for certain traits also, but actually they control the sex of a human being. So, what carries the human genome from generation to generation? We know that the children are produced due to the marriage of their parents, and so both parents, the father and mother, share together in order to pass their characteristics to their children. We'll find that a single genome, as we said, which is the 23 chromosomes, 22 autosomes, and sex chromosomes, which is haploid. We call it haploid. Why? Haploid because each type of chromosomes, type 1 chromosome, type 2, type 3, type 4, and so on, is found as single chromosome. And so we call it haploid set of chromosomes, okay, containing the 23 single chromosomes. 23 single chromosomes in a spermatozoon will find that there are 22 autosomes, the 22 autosomes, and either Y chromosome or X chromosome. However, in the X cell, we'll find that it has 22 autosomes and X chromosome only. It doesn't contain, there are no X cells with Y chromosome. And as we will see, the determinant of the sex is the spermatozoon. Now, how are these genomes carried from parent to their children? The transmission of traits occurs due to what we call fertilization. In fertilization, we'll find that a sperm nucleus, which contains the nuclear genome, gets into the egg and fuses with its nucleus. And so the 22 or 23 chromosomes of a sperm, together with the 23 chromosomes of the egg cell, together will start to form what we call the zygote. The zygote is one cell which contains the chromosomes of the father and chromosomes of the mother together. Now, as you see here, each type of chromosomes. For example, chromosome 1 is formed of two chromosomes, one from father and one from the mother, one from the father through the spermatozoon and one from the mother through the egg cell. The same thing with chromosome number 2 is also 2 or pair, and 3 is also pair. And so all the chromosomes are paired. The paired chromosomes in the cell or the zygote makes it a diploid, having a diploid nucleus. Diploid double, 2 means we have two chromosomes of each type. So the zygote contains two genomes, 23 pairs of chromosomes, one paternal from the father and one maternal from the mother. The zygote can either be XY, means male, or XX, female. How about the mitochondrial genome? We'll find that the mitochondrial genome is inherited from the egg cell. Why? Because the spermatozoon transmits only the nucleus to the egg cell. But the cytoplasm of the egg cell containing the mitochondria forms the cytoplasm of the zygote, which will form the embryo and the total human being. So, mitochondrial genome is maternally from the mother inherited from the mother, not from the father. Now, from one cell zygote to multicellular embryo, we'll find that the one cell will start to divide into 2, 4, 8, and so on. This division is called mitotic division or mitosis. By mitosis, the cells increase in number and the cells differentiate. Differentiate means what? Means the functionality of the cell is in one part is different from the other. That's why they form the tissues and organs of the human being, which also is formed of cells. And as we know, there is a continuous growth by division, by mitotic division, in order to increase in size, and continuous differentiation of these cells. And so the growth and the replacement of dead cells in the human body are formed by mitotic division. Usually, the cells of the body, we call them somatic cells, which can divide by mitotic division. Any cell which will get into, is destined into division, has to pass what we call the cell cycle. In the cell cycle, there are two main phases. The first is the division phase, during which the cell divides into two cells. The second is the interphase, during which the cell prepares for the next division. The interphase is divided into three phases. The first phase, during which the cell is destined or gets the information to start division is called the G1 or gap 1 phase. Why we call it gap? So it's in between two phases or two processes, and we will know that these two processes are the division of the cell into two, and we can see this under the microscope, and the other part or the other phase which we'll talk about is the formation of new genome, new DNA, in order to form the new cell. So this is the G1 or gap 1 phase. During gap 1 phase, the volume of the cell increases and all organelles replicate in order to be able to form two cells. Now, when this is completed, we'll find that the cell will enter into what we call S or synthetic phase. During this S or synthetic phase, the DNA and centrioles, which are important for the process of division, replicate, becomes double. So the DNA, if we have the DNA to form the genome, one genome, here we have double the amount of DNA. But remember, although it will form chemical material, which is DNA, but it will be exactly like the genome which is found in the cell. So the DNA is duplicated and the centrioles are duplicated. When they are duplicated, they start preparation for the cell division. And this occurs by synthesis of enzymes and proteins and so on that is needed for division in what we call the G2 or gap 2 phase. So we have two important phases. The first phase is the division phase, which we can see under the microscope. The cell is dividing into two. And the second is the S phase or synthetic phase, which we can identify biochemically and histochemically, in which DNA is duplicated. Between the division and the start of S phase, this is gap 1 phase. Between the end of the synthetic phase and the start of cell division is the second gap or gap 2 phase or G2 phase. Okay, now the cell is ready for mitotic division. Okay, what happens in mitotic division? The first phase in mitotic division is called prophase. During prophase, we'll find that the chromatin, which is formed of long fibrous material, condenses and forms the chromosomes. At the same time, we will find that the nuclear envelope starts to disappear and the nucleolus also starts to disappear. The centrioles begin to move to the opposite poles and form in between them the spindle fibers. These spindle fibers attach to the chromosomes in order to pull them and separate them during division, as we will see. The second phase is metaphase. Here the chromosomes are well packed and formed and appears as chromosomes. But each chromosome is formed of two sister chromatids. Why two? Because during the S phase, DNA has been duplicated or replicated. So the DNA of the original cell became twice as much as the DNA. So each chromosome DNA became twice the DNA of the original chromosome. This DNA, which is newly formed during the synthetic phase or S phase, appear as what we call the chromatid. Tid means twin. So the twins, the two twins, two chromatids, form each chromosome. So chromosome 1 is formed of two chromosomes, one paternal and one maternal, but each of the two chromosomes became two chromatids. Okay? Now, these chromosomes will line up at the middle of the cell and attached to the spindle fibers were attached to the centrioles. Now, we'll find during anaphase, the sister chromatids break apart and move to the opposite pole with the help of spindle fibers. And the newly separated sister chromatids are called chromosomes now. You know that it was two chromatids. But when they are separated, each of the chromatids became chromosome. Okay? And so these will separate from each other. In telophase, the chromosomes decondense and become in the form of chromatin fibers again, and the spindles start to disappear. Two nuclei form in a cell. These cells, by what we call cytokinesis. Cyto means cytoplasm. Kinesis, movement, so the movement of cytoplasm. And by the way, the prophase, metaphase, anaphase, and telophase are called karyokinesis. Karyos means nucleus. Karyokinesis, movement in the nucleus. Now the movement will occur in the cytoplasm by cytokinesis, and so the cells will be separated, and the one cell will have again two cells. Each cell have the same amount of DNA and the same number of chromosomes and the same nuclear genomes of the original mother cell. So we have two daughter cells now. Now, we'll find that as we said, the gamete, which is spermatozoon, or the egg cell, contains only a haploid number of chromosomes. That is, half the number of chromosomes of any cell in the body. This half number or haploid number is formed by a special process called meiotic division. The other was called mitotic division. This is called meiotic or reduction. Reduction because the two genomes are separated and only one genome will be found in the cell. In meiotic division, we will find that before meiosis, the cell passes through interphase G1, S, and G2. And so, before meiotic division, the cell will contain double the amount of DNA which is found in the mother cell. The number of chromosomes in the parent diploid cell, the parent diploid cell of the gamete, is called gametogonium, mother of the gamete. It has diploid numbers, two chromosomes from each type, from 1, 2 chromosomes, 2 chromosomes, and so on. So it's diploid. And this diploid has to become haploid, means that contains only one chromosome. The gametogonium have the number of genomes in the gametogonium has to be halved in the gamete. It produces, therefore, 4 haploid gametes. Remember that the cell, the somatic cell, the body cell, when it divides by mitosis, it was a diploid cell and gave 2 diploid cells. But here we'll find that the diploid cell, gametogonium, will synthesize DNA during the S phase and prepare for the meiotic division that will end with haploid gametes. And so the one cell will not give only 2 cells, but it will give 4 cells. And it will give 4 cells which are either, if we have the spermatogonium, it will give 4 spermatozoons, or oogonium, it will give 4 cells, 1 egg cell, and 3 small cells which are not egg cells. Now, meiosis comprises 2 cellular division, and so instead of having just 1 division, mitotic division, we have here 2 divisions in order to reduce the number of chromosomes. We call these 2 divisions meiosis 1 and meiosis 2, or meiotic division 1 and meiotic division 2. If we take meiosis 1, the prophase of meiosis 1 is a long process that, for example, in female, it starts during uterine life and it ends at the old age. And so the first prophase, or the prophase of meiosis 1, passes through 5 different stages to prepare for reduction division. Now, the first phase of prophase, of first prophase, is called leptotene. Lepto means fibers. Leptotene. And in leptotene, we will find that in leptotene, the duplicated chromosomes start to condense. This chromosome, the red, is actually formed of two chromatids, and the blue is formed of two chromatids, and they start to condense, liptotin. The second is called zygotin. The zygotin, zygo means to form two together. And so the chromosomes will synapse, that is, they will become around each other, like as you see in here, and so these synapse in the zygotin. Then the third is the pachytene. In pachytene, the synapsis is complete, and each chromatid takes a part of the other chromatid. The paternal, the chromatid from the paternal or the father takes a piece from the mother. So they exchange pieces between each other so that the children will have the characteristics of both parents. Now, the fourth is called diplotin, and in diplotin, the synaptonemal complex disappear and the chiasmata are visible, and so the chromosomes are attached to each other. Then finally the diakinesis, and here in the diakinesis the bivalents are ready for metaphase. Each chromosome is separated from the other chromosome. Yes, they exchanged pieces. You see that the blue chromosome exchanges a piece of the red chromosome, and so it has a piece of red chromosome. For example, if the blue chromosome was the chromosome from the mother, and it carries two characteristics of hair, for example, blonde hair and curly hair, will find that instead of being blonde and non-curly, as the other is black and curly, the child will have black hair but non-curly hair. Okay. First meiotic division. The late prophase, the pairs of homologous chromosomes form tetrads. Why tetrads? We said that we have a chromosome from the father and a chromosome from the mother, but the chromosome from the father has two chromatids, and from the mother has two chromatids. 2 plus 2, 4. 4 means tetrads. Tetra means 4, as yani twins, and so they will appear as 4 twins, with crossing over, of course. In metaphase, we'll find that the homologous pairs of chromosomes align on equatorial plate. Look here, that the chromosome from the father with its two chromatids and the chromosome of the mother with its two chromatids are paired together. Okay? So in anaphase 1, the chromosome from the father with its two chromatids will separate from the chromosome of the mother with its two chromatids. And so half the number of chromosomes will be on the left and half the other half on the right. And this is the reduction of the number of chromosomes. So each chromosome with its two chromatids, the paternal chromosome with its two chromatids, will be separated from the maternal chromosome of the two chromatids. When they form, as you see here, that the two chromatids are together of the paternal and maternal, okay? And each of the chromatids exchanged pieces with the other. So there is a piece in the paternal chromatid from maternal and piece of maternal from the paternal chromatids. Now, in telophase, we will find that the paired chromosomes are separated, each with two chromatids, yes, but the chromosome itself is chromosome 1, just one chromosome with its two chromatids at the right, and one chromosome with its two chromatids on the left. So the number of chromosomes becomes half the original number. Okay? Now, in cytokinesis, two cells will be formed, but each cell is now haploid, having half the number of chromosomes. Okay, each of these cells will start again to divide. And the division here will be very similar to the mitotic division. That is, each chromosome with two chromatids will appear, and it will line in metaphase 2 at the equatorial plate, and then when it is pulled, the chromatids will be separated in order to form finally four. You see that the chromatids will be separated, and so four cells, each of the four cells have the half number, or the half number of chromosomes, or in other words, each is haploid. And these four cells form the gametes, which are either spermatozoon or egg.